amazing to be on this platform. And thank you so much to the LCA team, but also everybody joining tonight. And it's so wonderful that the ocean secured a whole day to itself, which is World Ocean Day, of course, today, declared by the United Nations in 2008 only, so not so long ago. But yeah, it's dedicated to celebrating what it means to us, what the ocean means to us as humans. But like Aisha said, also to highlight the ocean's importance, but also fragility, and reminding us all to be custodians of this incredible environment that covers 70% of our planet. So as SAAMBR, we celebrate, we live, we breathe, not always eat, but we research, we love, and we share the ocean 365 days of the year, while working really hard towards inspiring people to connect with the ocean in a very positive way. Now, like I mentioned, we realized that I rarely actually introduce myself. So here we go. So besides all that stuff that Aisha said, I'm actually just a straightforward, what I call ocean frother, ultimate fish nerd, yes. And more recently, I also tend to have meetings with pythons, which I'm very proud of. But actually, I'm just surrounded by incredibly passionate people, and some of them will be joining us tonight. So it's going to be a great evening. And most of you might remember the old Durban SeaWorld, and that was built in 1959. And 8 years after SAAMBR, which is the South African Association for Marine Biological Research, was established. So that makes us 72 years old, which I would say is mature and mostly wise, I hope, but also still full of fun. And our mission back then, and still now, is to contribute to conservation with a very deep foundation in science and research, which we share through education and awareness activities. Now, the facility moved to the Point area 18 years ago, and we now, this beautiful green oasis in quite a rundown urban area. And we also play host to bees and bats, by the way, not just fish. So it's really creating an ecosystem, a natural, beautiful space for people to visit. So we have 3 very prominent divisions covering research, the aquarium, with the purpose of growing ocean awareness and, of course, education. And, you know, that in combination really covers all the bases of the ocean and coastal areas. So the Oceanographic Research Institute, or what we call ORI, is world-renowned, with about 50 current research projects, and mostly focusing on the Western Indian Ocean and also coastal areas. And then the aquarium, it is the biggest aquarium complex in Africa. We've got 22 million liters of seawater, and it's home to more than 510 species. And we see hundreds and thousands of mostly local visitors. We really do amazing work. And I have to give a big shout out to our friend Don, very special photographer, who's been taking these incredible images of our work that we can share with people. And yeah, he's 100% part of our family. So thank you, Don. And then our education team, they make real world impact. We cater for learners, all sorts of learners, from the littlies in the early childhood development phase, all the way through to different able children. And of course, the teens and the young adults. And older adults. We also do, you know, postgraduates and mature students. And we offer interactive, experiential, environmental education absolutely at its best. And none of this, of course, is possible without our much loved volunteers. So always a massive thank you and appreciation to all the volunteers who help us to achieve our mission. So with this fantastic mix of ocean people and creatures working towards connecting people with the ocean, in all sorts of different ways and with all sorts of different stories and signs and just inspiration. So I have this evening a wonderful team of presenters who's going to join us and share what we do on a day-to-day basis. And we're going to start shortly with Dr. Ash. Now, she loves what I'm just going to call, most people will recognize as sea hojas. Then we're going to move to the wonderful Nomfundo, who inspires kids every day. And then we're going to listen to Dr. Justin, and he will show us some coral porn tonight. So please stay tuned. And then, of course, we have Ryan, who saves sea turtles, and it's amazing. And then we'll wrap it all up with Nikhil, who's our communications champ, and he makes sure that we share science-based knowledge with all our audiences. So here is SAAMBR, and Dr. Ash is going to join us, and we're going to switch over to that screen. So thank you very, very much.
Hi, everyone. I'm Dr. Ash, and today I will be talking about our research division known as the Oceanographic Research Institute. It is a division of SAAMBR who strives to stimulate community awareness of the marine environment through education and to promote wise, sustainable use of our marine resources through scientific investigation. Much of the work of the institution is focused along the KwaZulu-Natal coast, and we are one of the leaders in the Western Indian Ocean in terms of research, and we have worked on projects in all countries around the Western Indian Ocean, including the island states. So, ORI is made up of 6 different portfolios, starting with the demersal fisheries and ecology, the coral reef research portfolio, the estuarine and soft sediment ecology portfolio, and the coastal zone management portfolio. I am, however, part of the regional fisheries and invertebrate fisheries portfolio. So what do? I'm a postdoctoral researcher here at ORI. We jump on different ships and we go and collect, like Mareka said, the jojos of the ocean. So basically it is zooplankton, the little babies in the ocean. So the reason for collecting these is we do a lot of genetic research on these zooplankton, which I will get into now. So why zooplankton? Why are we selecting these little jojos? These small organisms dominate the abundance and the biomass of the multicellular pelagic animals. They play an important role in the transfer of energy from primary producers, which are your plants. So our zooplankton eat up the plants, and this energy gets transferred to higher trophic levels, so your bigger fish, and so have you eat your zooplankton. So this is how the transfer of energy occurs. They are also mediators of the biogeochemical flux. And these little buggers are actually excellent indicators of biodiversity change as they respond rapidly to environmental change. So if there's anything going on in our oceans, we will see the change in these marine zooplankton. So we, in our team, use DNA metabarcoding. So we involve genetics, and we basically try to identify the different zooplankton that we collect. So like I said, they are highly diverse and highly abundant in the ocean. So there's millions and millions and millions of zooplankton whenever we go out to sea. So we collect these zooplankton in these little honey jars, and we now need to identify all the zooplankton in these honey jars to a species level if possible. So we collect the zooplankton and we basically mash up all the zooplankton and we extract its DNA. And by this, we sequence this entire mashed up DNA using next generation sequencing. Once we have this and we have the results, we are able to identify to a family level, order level, genus level, but we try our best to get it down to species level, the different organisms that are present in our zooplankton mash, our little zooplankton pool. So the importance of this is DNA metabarcoding provides a cost-effective and reliable way for species identification, which is fundamental for our research and for future large-scale biodiversity monitoring programs. This tool is currently transforming our capacity to assess biodiversity. So, monitoring is an integral part of like marine ecosystem management. If we are able to monitor the marine biodiversity in our oceans, we can answer a lot of ecological questions. So that brings me to an example of what we have done in our research group using zooplankton and metabarcoding. So along the east coast of South Africa, within the Western Indian Ocean, we have the Agulhas Current. The Agulhas Current travels down towards, from all along the east coast of South Africa, joining Durban to Cape Town, and so have you. So the countries along the east coast of South Africa are also connected by the Western Indian Ocean. And the currents allow for the transport of organisms from tropical regions in the north to subtropical regions in the south, because these larval zooplankton are stuck in the water column in the pelagic region. So basically, you get your current that is pushing all your larvae down from the northern areas to the southern areas. So we use these marine zooplankton as model organisms to track, as I mentioned, what's going on in the ocean. So one of the projects that we did, we collected zooplankton all along the east coast of South Africa. And an interesting result that we saw was that we were able to identify 19 tropical Western Indian Ocean decapod species in KwaZulu-Natal waters. And why was this interesting? Because these zooplankton, the adult forms were not found in our KZN waters before. We have never seen the adult form of these zooplankton in our waters. And only through metabarcoding were we able to actually identify now that we are getting the zooplankton traveling along the east coast of South Africa via the Agulhas Current, indicating a southward advection of drifting life history stages along the east coast of South Africa. And this is great news, because there's future trajectories which have shown us that these adults, in the next 50 years, we should be seeing these adults settling on our benthic environments. And this is what we were able to see with metabarcoding. Thank you. I now call over Nomfundo.
Hi, everyone. Thank you for having me today. My name is Nomfundo Buthelezi. I'm working as one of the educators for SeaWorld. I'm very excited to be sharing with you today the role of Education Center in inspiring people to care for the ocean, or basically inspiring behavior change. Who is education? Ushaka SeaWorld Education Center is well known as the leading marine conservation education center in Africa, with over 130,000 learners visiting us on site, over 12,000 learners for off-site visits during our outreach programs, and obviously our day guests, which amounts to over 780,000, that is on annual average. Ushaka SeaWorld Education Center is aiming at provoking the behavior change on our guests by inspiring them to take care of the marine environment. So in order to efficiently perform this, Ushaka SeaWorld Education is operating in its two integrated subdivisions, which are formal education as well as informal education. So what does formal education do? Under formal education, we do have several programs that provide exclusive teaching opportunities that helps to keep the groups coming year by year, because it has been observed that the groups are coming back once they have visited us. And again, Ushaka SeaWorld Education is making sure that is providing these programs which are directly linked to the basic education school curriculum. Amongst some of the projects that we do on site, we do offer guided aquarium tours for different age groups, ranging from like the tiny toddlers to primary schools and obviously high schools. With the use of our venues, like our conference hall, our classrooms, we are able to also offer PowerPoint presentations that are accompanied by the hands-on activities plus the practical experiment. We also offer primary and high school courses to learners. They are able to choose the theme of their choice, depending or based on their curriculum requirements. So one of our regular courses includes the high school classification course, where the learners get the opportunity to touch and feel our animals of different phylas, so which are either live or preserved, of which there is something that is not available in class. Not everyone can afford coming to Ushaka. So with the use of our sponsorships, we also do school visits and off-site lessons. Sometimes we do visit schools and present to them some of our lessons, and sometimes we do take them to the actual field, because sometimes with the learners, when they get to explore the actual field, it just increases their perception. So when you're looking at this picture, you can see, so it's very like so heartwarming and amazing looking at how they will eventually fall in love with the sea creatures. We also do special interest groups, so which are organizations and tertiary schools. As you can see, we sometimes do offer them the opportunity to go to field to practice within their field of practice. And with our informal education, we do exhibit interpretation to guests. And also at the same time, with the use of our facilities like the aquarium, we are able to leverage on emotional connection between our animals and our guests while sharing animal stories. And with most of the stories that we do share with our guests, they are so like inspiring. As per se, science speaks to the brain, but storytelling speaks to the heart. So we hope to achieve and to change their perception about the animals by just giving them some really, really inspiring stories. So with both of the education departments, they do focus on the main three issues that our oceans are facing, which are plastic pollution, climate change, and obviously over-harvesting of natural resources. So we tailor-made our messages to suit our audiences. For an example, Simply not littering is an important message to one group, and avoiding clothes containing plastic fibers may be relevant to the other group. Both messages tackle the issue of plastic pollution facing our oceans. However, these messages must be relevant to the groups we are talking to. So in conclusion, we as Ushaka SeaWorld Education believe that the environmental education must be fun and relevant to our audiences. We work very hard to ensure that we engage optimism and empathy for marine creatures and our oceans to help our audiences rebuild connections to the natural world. Thank you.
Good evening, everyone. Happy World Oceans Day. I'm going to tone the lights down and say the over 18 sections just started. My name is Justin Hart, and tonight I'm going to be chatting to you about some of the work that I did for my master's and my PhD research based at ORI. I'm currently the aquarium curator, so I got a little bit tired of reading up on papers, and I've moved across ship to the aquarium side with a keen interest in looking at how we can get aquarium science up and running here. So my master's and PhD research was on coral reproduction. And I'm not going to go into too much depth on a Coral 101 course, but for those of you that aren't aware, corals are living animals. And there's a miraculous relationship between a algal species, a zooxanthellae, and a host, which is the coral polyp. This relationship is key, where the coral actually gets most of its energy from the algae that lives within its tissue. And at the basic level, you have a coral polyp, which then live together to form a coral colony. And coral colonies live together on a reef to form what we know as coral communities. And that's what you most likely see on your National Geographic photos. And what's interesting about these ecosystems is that they are the world's largest living structure. The Great Barrier Reef covers the extent of 2,300 kilometers at length. To put that into perspective, that's a flight from Durban to Cape Town and back worth of living animals made up by tiny little polyps. Pretty incredible. But yeah, from a distribution side of things, corals are primarily found within the tropics, where light levels and aragonite, well, carbonate chemistry supports the growth of accreting corals. They only cover 0.2% of the ocean floor, and it's miraculous that they actually support 25% of all marine species. So that's to just put things into perspective. South Africa, we're sitting at a high latitude, so our corals are different to what are known as accretive reefs. Our reefs don't grow towards the surface like the tropical reefs do. So instead, we have a thin layer of coral on ancient fossilized sand dune instead of compounding accretive reefs. Nonetheless, they are highly biodiverse. We've had over 93 species of hard corals, 39 species of softs, and in excess of 450 species of reef-associated fish described from our reef system. Now, getting on to the fun part, the reproductive cycle. If you look at, I'm just going to flip through just to put it all up into perspective. So a coral being a sessile animal, it's attached to the reef. It doesn't have an option to move down the reef to go find a mate. So what they do is the majority of corals broadcast spawn egg and sperm into the water column. And the fascinating part about this is that if you imagine you've got a friend down on the reef that you need to mate with, you need to get the timing right with that. So the synchronization for me still fascinates me, where the temperature cycle governs what month the spawning occurs. The lunar cycle governs what day the coral will reproduce, and the time after sunset governs the time that the coral actually releases into the water. So in many places around the world, like the Great Barrier Reef, you can have what are known as spawn slicks, where if you look from the sky, it looks like a red tide of sort. That's millions of gametes that are sitting on the surface, busy getting fertilized or developing into larvae. So they have a pelagic phase where they then get dispersed, and then they then have to find a suitable spot where they can settle. Now, once you settle, you also have to be happy where you settle, because you have to survive that location, and it's not easy down there. You've got competitors, you've got coralline algae that can overgrow the corals. So it's a very selective process when they actually settle back on the reef. Now, from an external point of view, you can't actually tell when the coral's ready to spawn. So what we do is we take biopsy samples, we take a small nubbin sample, and then you look at the cross section, and on the right-hand image, you can see that red pigment in the skeletal tissue, and that's actually eggs within the tissue of the coral. If it's pigmented like that, the general rule of thumb is it's likely going to spawn after the next full moon. So that way you're able to start tuning down exactly what day the coral is going to spawn. Now, the guys in the Caribbean get it lucky where they can swim around at night, doing night dives and put nets over corals, have a cup of tea if they want, but not us in South Africa on a high energy coastline. We have reefs that we cannot dive on any night that we like. And for that reason, we don't know much about the reproductive cycle of corals, because it happens under the cover of darkness. So what we do here is in the Ori Research Aquarium, which is fed by water that's supplied to Shaka SeaWorld, we would collect representative samples of a coral, bring it back to the research aquarium, and then I devised a bucket system where every night I'd go in and put a bucket over a coral. And then once the coral's spawned, it will then release the gametes that float up into the surface, and then I can collect those gametes and do a variety of designed experimental cross-fertilizations on that. Now, this is a video that it's the first time it's being shown, but this was taken at the research aquarium on one of the species that I was working on. It's a staghorn coral called Acropora austra. And what you see is actually live of what actually is happening with the spawning in real time. It's not slowed down or sped up. So what look like polystyrene balls are actually an egg sperm bundle that's been released by each of the polyps. And that floats up to the surface, and each of those bundles in this species is about 32 eggs packaged around a parcel of sperm. When it gets to the surface, it then has a time where it dissociates. Obviously, this is a vital food source, and you can see even a barnacle over there trying to get his fair share of bundles coming past. But once they get to the surface, they then take about 15 minutes for those bundles to actually break apart. And only then can cross-fertilization or fertilization take effect. So that's just a clip of what had happened in the research aquarium. Now, you can imagine this is a stressful thing, because if you miss it, you come back next year. Then once the larvae have developed, what you see on the left-hand side is one of the larvae that I had on a settlement plate in the lab. And because we cannot study this on the reef because of the logistics, being able to look at the settlement dynamics is key in a lab environment. And with a bunch of experiments that I did, I showed on how selective settlement can be. And there's a little coral on the right-hand side, or 3 that have decided to settle in the same spot. And that's before they've even started secreting a calcium carbonate skeleton. The row of images at the top show a coral that's 2 days old. And you can see there it started to secrete some skeleton. All those ridges are all skeleton. And then after 15 days, it's starting to take on the zooxanthellae, which you can see that brown in the polyp tissue. And on the right-hand side, that's pretty much a month later. Now, what I also did is in the wild, on the reef, I put down settlement tiles, and those were returned to the laboratory every 6 months. I kept them alive. And then I was monitoring the dynamics of how those corals responded to benthic stuff that was surrounding them. Then came the part where I got involved with the Shaka Aquarium, where after learning how to monitor these or predict when coral was going to spawn, I then went to the main coral exhibit and then started to survey the corals there. And I found that one of them had gametes that had developed. And then I put the net over the coral, and on the top right-hand side, you can see the gametes that were collected on one of the nights. In excess of 312,000 eggs were released by a single coral that night. Unfortunately, we didn't have another a mate for it, so I couldn't cross fertilize it. But on the left-hand side, that is not a smoothie. That's all the eggs that were collected. And that was the coral. On the bottom left is a Cropper oppressor. And at the bottom right is just an image of the eggs. So in a nutshell, from what the work that I'd done by moving into the aquarium space and using the facilities at SAAMBR, I was able to look at these vital life history stages, starting to get a little glimpse on an essential process that we don't know much about. And we found that the spawning happens after the February or March full moon, and that the captive breeding techniques that I was learning and that other aquariums are also working on is going to help restore reefs in other places of the world. If you do want to find out a bit more, there is a paper that was published on it in Reef Encounters. And with that, I'm going to pass over to Ryan for some turtles.
Hi, everyone. I'm Ryan Rombrun, and I work in quarantine and in the Turtle Rehabilitation Center at Ushaka SeaWorld. And what I'm going to be talking to you about today is basically a little bit of history, a little bit of biology, because I am a biologist at heart, and then a little bit more about our rehabilitation that goes on here at Ushaka SeaWorld. Okay, so South Africa has a very rich turtle conservancy history. We have 5 of the 7 global species that occur in our waters. So we have 2 nesting species, which are the loggerheads and the leatherbacks, and 2 foraging or feeding species, which are the greens and the hawksbills, and 1 vagrant species, which is the olive ridley, which neither nests or forages here. So we actually have the 2nd longest running sea turtle monitoring program globally, which was initiated in 1963. And as Marika said, this year marks 60 years of sea turtle monitoring in the country. And SAAMBR as an organization has been affiliated with this conservation from the initiation of this conservation, with George Hughes, who's often referred to as the father of sea turtles. And he was actually a student here at SAAMBR, and his PhD, which was completed in 1974, covered the sea turtles in Setswana. And if you want to read more about that and his adventures, you can always, he wrote up a book, so you can always purchase that, and it's called Between the Tides. Okay, so you can't effectively conserve and manage a species without understanding it. So I am going to give you a crash course in sea turtle biology, and it's a very complex one. So it starts off on the beach as a little sea hatchling. They usually get incubated for 60 days. And then they are hatched and they make a wild scurry for the shore. And it's almost a 2-day journey. So they swim for the reef, and then they swim past the reef. So the reason that they do this is because 90% of your predators or 90% of your fish are actually found in your coastal zone. So as a strategy, they swim out into the oceanic realm to avoid predation. Here, they'll spend the next 10 years of their life out at sea before coming back to the coastal areas where they'll start feeding again, and they'll develop their species-specific diets. They'll stay here until they reach maturity. This can include, depending on the species, between 10 to 36 years. And then they undergo breeding migrations to their nesting sites. Once the males are done with copulation, they go back to the coastal foraging zones, and the females will actually stay behind in the nesting site where they'll lay multiple clutches of eggs throughout the season before also returning to the coastal feeding zones. I also want to highlight that to get to the actual breeding stage, for a female, only 1 in 1,000 actually makes it to that stage. Okay, so why are sea turtles important? Why should we conserve them? So sea turtles act as flagship species. So these are important because they make people feel connected to the ocean, and you wouldn't have an Ocean's Day without a turtle, someone thinking about a turtle. So people care about turtles, and they are synonymous with the ocean. So people care about the ocean. And people protect what they care about. Secondly, sea turtles fulfill various ecological roles in the ecosystem, which is important for certain ecosystems to persist and to be healthy. And then I just want to tap on one of the roles, which is habitat modifiers. So Justin spoke about your coral reef systems, which are in delicate balance with algae. Not only algae within the animal, but also algae around the animal. So algae, funnily enough, competes with coral and will often overtake a coral reef. So it needs to be managed. And it was found by a study in 2012 that actually greens and hawksbills are 2 times more effective than reef fish in maintaining these coral reef systems. And in South Africa, they are certainly doing that job, because in green turtle gut content, species diversity of 31 different species of algae were found in the stomach contents of greens. So they are certainly helping maintain the coral reefs in our region. And then I also want to highlight that populations are distinct to the region. So what I'm showing here are the 4 species that we find in our water, in our waters in the Indian Ocean, and each dot is essentially a nesting site. So the bigger the dot, the bigger the nesting site. And if a population goes extinct, it's basically a local extinction event. So you cannot repopulate that area, and you lose those ecological roles in those areas and debilitated ecosystems as a result of that. So now that we know why we protect them, as I said, SAAMBR has a rich history of sea turtle conservation. It started in the 60s, and in terms of rehabilitation, it goes all the way back to the 90s, and we've actually rescued over 400 turtles with a success rate of 80%. And we are the only rehabilitation center in KZN and one of 3 in the country. So I'm just going to talk you through the rescue, rehabilitation, and release process that happens at our facility. If you do come across a stranded individual along the eastern seaboard of South Africa, you can always contact us on the numbers that you see on screen, either during office hours or after hours. It's a 24-hour line. Sorry, and usually your first responders to that will be someone from the KZN Stranding Network, which is basically a collection of amazing and passionate individuals from various facilities and institutions who come together, and they are basically, as I said, the first responders, our on-the-ground people who will respond to you and collect those animals and bring them to our facility. And then I just added in a couple points, general points, not to, if you do come across a stranded turtle, that you don't put it back into the sea. And you don't attempt to move that animal until a member of the network is with you. And you never place the turtle on its back or hold the turtle down on its shell by pushing it down, because the lungs lie directly under the shell. So essentially you might be suffocating that animal. Okay, then we get to the rehabilitation part, which is what we do at the facility. And I am part of an incredible team that works to help these sick or injured individuals. And what we would do is we basically, when the turtle comes in, we have a primary assessment where we record weights and measurements. And then depending on the severity of the case, we either allow that animal to rest or we run diagnostics. Diagnostics will include things such as bloods, radiographs, and ultrasounds. And then from there, we will look at the results and we'll basically draw up a treatment plan. And it's a constant monitoring and assessment process that happens. And into that we feed, it comes into husbandry and good nutrition. And then we rerun diagnostics, and we essentially determine if the animal needs extended care in a managed environment or if that individual becomes a suitable candidate for release. Once animal is cleared for release, we will determine a suitable release site for that animal, which will be dependent on the species and the life stage of that animal. And we will thereafter tag that animal, so a flipper tag in both front flippers, a microchip, and then if we are lucky enough, we do get to satellite tag these individuals. Satellite tag and acoustic tag these rehabilitated turtles. So these tags are very expensive. So it is always opportunistic, and it's ideal to look at it because it allows us to monitor the journey of our turtles post the rehabilitation. And we can assess behavior, so we can see if our turtles are behaving as a wild turtle would, and also the survivorship of our turtles. And it adds also to the movement data of South African turtles, and also contributes and makes us see the bigger picture of how our contribution is made to the sea turtle conservation program. Cool. I'm now going to hand over to Nikhil, who's going to take you through communications.
Thank you very much. Happy World Ocean Day, everybody. And let's get this going. So yeah, my name is Nikhil Singh, and I do some of the communication work at SAAMBR, which is basically the idea of taking all of these incredible things that Dr. Ash, Dr. Justin, Nomfundo, Ryan, Mareka have spoken about, and getting that into a format that people can understand and hopefully change a behavior based off it. So I end up getting information that looks like this. Pretty, isn't it? It's a table with a whole bunch of information. And when you look at something like this, it doesn't exactly draw you in. Most of us look at it and don't really know what we're looking at. It's my job then to take this information and turn it into something like this. This will tell people about those species, how many of them we've had. And like Ryan mentioned, we've had over 400 of those animals through our doors in the last few years. Now, apart from that, apart from the idea of just taking the information and translating it, it's also about the ability to get people inspired. The way to inspire someone is to empower them. So we've also created an online platform, the SAAMBR Campus, and that allows people to take little online courses about conservation, about behavior management, and so on. Small animated courses that will spur that need for conservation within everybody. Okay, now another cool perk of the job is the ability to host things like this. If you haven't seen it already, head over to Durban's Promenade and have a look at the turtle exhibition. This was for World Turtle Day, and we had an incredible display of all of this artwork up, as well as some information to inspire people that are taking that morning stroll and just need a little bit of a rest. They can sit in front of this board and learn just a little bit about sea turtle conservation. When someone comes to a facility like this, there's always this sense of wonder and this sense of awe. And we try to use that sense of wonder and drive home a conservation message. It's why platforms like this and facilities like ours are so important in the conservation world. They drive that behavior change, and communicating those points to them is so, so vitally important. So from all of the speakers that you've heard today, I can personally say that I'm feeling inspired. I'm feeling like a better conservationist just from being in their presence. They inspire me on a daily basis, and that's why I'm here. We're going to break now for some questions and answers. All right, so we'll start off with the panel, Aisha, yeah, and then she'll host that from there.
I want to say actually thank you to all our speakers tonight, for very informative and very fascinating talks. Today when I'm based in Cape Town and it's on the cold Atlantic side, and today when I left work, it was storming. So I just passed by the ocean and I sat in the car because ironically I didn't want to get wet. So I just sat there, and that's how I celebrated. So this was a fantastic way to celebrate it tonight. There are some questions in the chat. But before we go there, Chris, if you don't mind, I'm just going to ask a few questions that I have myself.
Of course, welcome and listening to your questions and seeing, also want to hear the answers, but thanks to the whole team. Just want to say that, we say in the hospitality industry, passion sells, and your passion is really, really something that strikes me. And I thank you for the absolute dedication of the team and the incredible work you do in a huge ocean and a huge task. And also thank you to the sponsors behind you. I think this is something that we all should know more of, learn more of, and get involved more. Thank you to a wonderful, passionate team. Over to you, please, Aisha.
Okay. Marika, thank you for answering so many of the questions in the chat already. So I'm going to go with my own questions, excuse me, and then we're going to open the floor for more questions. So my first question is directed at you, Dr. Ash. To me, it appears as if ocean science research is starting to include more and more high-tech equipment and modeling. I think it's fair to say that. So in future, do you see more, let's call them tech geeks, contributing to research and conservation?
Yes, I do think so. And I think that it's already happening, especially in areas like DNA sequencing and artificial intelligence. They are already allowing us to understand the natural world and conservation in wildlife in a more efficient and effective way to allow us to safeguard species extinction. Even in my own work, we use a lot of technology, a lot of equipment out in the ocean. We're using next generation sequencing that's allowing us to readily and quickly identify species, which will help us for future conservation efforts and to prevent like species that we know that are going into extinction, to allow us to conserve these individuals.
Okay, thank you. Yeah, we definitely have to embrace technology for the future. The next one is directed at Nomfundo. So Nomfundo, have you seen a shift in the children's awareness about the ocean? Or do you mostly see children who have not yet been exposed to this wonderful world?
I will consider this question as a 50-50 situation. We do get regular visitors, occasional visitors, and first-time learners. So sometimes with our regular visitors, we do see the shift, because with most of them, they seem to fully understand the link between where they stay and the ocean. But to your surprise, at some point, we do get the learners who are visiting us for the first time, but only to find out they are very clued up about the ocean and what's happening. And vice versa, at some point, you may find the learners that are just staying nearby the coast, but they do not really understand the importance of the ocean. They just realize the ocean as just water full of salt. But I think there are other things that we need to consider, like the type of school, the geographical location, and also what they are maybe taught to watch at home. Because sometimes with the kids that haven't been to the real ocean, but sometimes they do watch channels like the geographical, what's the channel? National. Sorry. Yeah, that's it. So it's just like the matter of 50-50. Sometimes there is a shift, so it's very difficult to measure. Thank you.
Yes, thank you. I'm just thinking, I'm just going to speak about the personal experiences that I grew up in Kimberley, which is inland. And yeah, my passion was so in that I needed to get to the ocean. So I completely get where they're coming from. The next one is for Justin. Justin, fascinating story about coral lives. How do you translate coral biology to teaching people about climate change?
I think the documentaries are doing a fair job at that already. Just with coral bleaching on a global level, it's received a lot of media attention. Most people that have seen a coral reef have also seen a degraded coral reef, where that association that I explained with that symbiotic relationship between the algae and the host, that has been broken down, where the algae is actually being expelled under a stressful condition. And people do make that link when you can explain to them on that symbiotic relationship and what happens when that relationship gets broken, because once that zooxanthellae has left, the result is pretty much a transparent host that shows the skeleton below it. So the proof's in the pudding. As soon as the bleaching has occurred, people can relate what the biological process that's occurred.
Aisha, you are muted, please.
Sorry. Thank you, Justin. I don't want to take up too much time, so I'm just going to go with one more question for Ryan. Ryan, of course, we all love turtles, and I want to know, what did you say is the single most important action that people can take to keep the ocean turtle friendly and in that way also healthy?
I think something that everyone can incorporate into their life would be the use of eliminating single-use items, because these often find their way back into the ocean, and these animals can't really differentiate what's food and what's not food, and they might ingest that. So I think it's also an effective strategy, because we've seen the elimination. I mean, we don't even see plastic straws anymore. It's rare. So I think just eliminating or reducing single item use should be something that everyone can do.
Thank you, Ryan. Yeah, I think whenever we speak about pollution or we speak about climate change, yeah, the coral, we always see coral, and like you said, with the turtles. I think each one should just make an effort now. I am going to go to the queue. Let's go to the floor first. Marit, are there any hands up?
I don't see any yet, Aisha, but yeah, I'm sure they'll start popping in now.
Okay, then there was a question in the chat where, yeah, Mareko already answered this, but I'm just going to read it. It's Don Hunter asks, can cross-fertilization take place between polyps that are adjacent to each other in a coral structure, or are they related? And Mareko answered that corals grow by asexual reproduction of polyps. Therefore, adjacent polyps are clones. And fertilization between adjacent polyps would therefore be self-fertilization, which is possible, but occurs only as a last resort. Yeah, no, okay, it is possible as a last resort if cross-fertilization is not possible. I don't know if you want to add on to that, Mareko, because if not, then I'm going to ask
Well, Don is
Tawani.
actually, I think, in the audience, so he can ask more questions, but Justin actually answered that because he's the pro and he can add to that. So Justin.
Okay.
Just relating to that, Don, what I was mentioning with that that spawning that happened in the aquarium, after with not having a potential mate for it, I looked at how I actually I separated the egg and the sperm away from each other. And then I did a selfing experiment where over time I started introducing the sperm back to the eggs. And only after about 3 hours was fertilization starting to come into effect. And it only reached a maximum of 5% fertilization. So they go through a strategy, and it's well covered in the literature, that the barriers to self-fertilization from the egg side. And then after a certain time period, they do reach a point where those barriers break down, and it's a case of beggars can't be choosers, and they can actually self-fertilize it to an extent.
Thank you.
Thanks, Mareko. Don, you want to come in?
No, question answered.
Okay.
One further part to that would be, does Justin know what the primary objective is? Is the primary objective to spread genetic biodiversity, or is it just to continue the species?
Sure, that's a tough one. It's a tough one, Don. I mean, based on the amount that gets released, yeah, they need to produce it. It is a very it's an energy expenditure that they have to do to produce gametes, but just by the vast number that actually has to be produced. Yeah, the dispersal stuff, we don't really know much about the theory behind the strategies that they're opting for, but it's very much put everything out there as much as you can. If you look at the spawning events, there's only what can actually happen is with a spawning event, you might have the first night that they do actually release just a little bit of eggs. And that threw me off quite a bit because I was running for my lab protocol to get going, and it literally the corals just released a little bit. But the second night following that, generally they just go off and put all eggs in one basket as much as possible.
Okay, then the last thing I want to throw in there is then, is the growth of the fertilized animal now,
yeah,
when it lands on a coral structure, is there competitiveness there? Do the local corals allow it to happen, or do they try and kill it, or do they facilitate its integration into the colony?
No, they won't facilitate it, Don. It will be there's a level of allelopathy that will happen, and that's why we don't know the literature on how much actually makes it through the recruitment cycle. So we know that from a settlement side, all lab-based stuff is that it's a very selective process. And based on what I've done in the lab as well, by seeing the amount of stuff that actually gets out-competed, it's a tough life out there. I mean, what I showed with that one picture where 3 had actually settled next to each other, that's a strategy that throws another spanner in the works to the whole genetics of corals, because what those could be is 3 different genotypes that have actually settled next to each other, and they form what's known as a chimera after that. So those would then keep on reproducing asexually, and then genetically on a coral, you could actually have 3 different genotypes on one coral. But the theory behind that is that might be a strategy that they're actually trying to grow as fast as they can. So find a mate that you can actually, or a colleague that you can pair up with and get through that bottleneck of the post-settlement success.
Thank you, Aisha. I think Tawani Nodia has got a hand up for a while. I'm going to ask to unmute. Please, Tawani.
Hi, can you hear me?
We can hear you, thanks.
Okay, perfect. Sorry, my internet is not good enough for me to have video. First, I would like to say thank you guys so much for the talk tonight. It has been fascinating. I've learned so much. It's really cool. And then my question is also to Dr. Hart. I've seen a little bit of the corals in the Mediterranean Sea, but I'm assuming that the structure of the coastal area in KwaZulu-Natal is different than it is in the Mediterranean coastal areas. So my question, I have 2 basically, is how has the negative effects of climate change affected the corals in the KwaZulu-Natal area? And does the sediment type and the way that it's formed make a difference in protecting the coral reefs in South Africa at all, or not so much?
Cool, thanks for the question. So relating to that, the South African reefs are actually considered to potentially be a refuge, in the sense that our reefs are generally deeper reefs, so they don't break the surface. They reach a minimum of around 7 meters on some of our shallower reefs up at Sudwana. And with that, we do have an element of protection from the shallower reefs that would be exposed to high temperatures in the tropics. So we do have bleaching events, but they're actually, I think that it's more defined as paling events, because what happens is our corals generally recover. So with bleaching, the corals can die if that stress event is too long. We do have paling that is recorded on our reefs, but generally those corals do recover after the stress, because it's usually very, very short-lived for us. So in that sense, our corals, there is quite a bit of interest elsewhere. We do also have some deeper reefs as well, and the whole topic on mesophotic reefs or deeper reefs on being refuges as well is also the case for us as well. But yeah, from a sediment side, our reefs are generally, well, we don't have much river inputs up at Sodwana and Isimangaliso on the northern sections. So our reefs generally don't get silted over, but we do have high storm events. So we do have a lot of breakage from big storms that come through, which then is requiring recruitment to actually come and replenish damaged coral tissue.
Kawani?
Awesome. Thank you so much. And then I have one more question about the collection of the plankton that you guys do. You have a tendency of sorry, in the first part of the talk, you guys said that you match them all together. So basically, you kill them to identify DNA strands. I'm assuming that you do this. It's an easier way of doing that than using like microscope to identify what they look like. But I would like to understand how different it is if you use a microscope and do, like with normal species, you know, you can take photos and identify species that way. But I'm assuming that that's a lot more difficult when you do it on such a large scale with so many different varieties in such a small sample.
Okay, so like you mentioned, we have these samples, and there's like thousands and thousands of zooplankton, right? And it's highly diverse. The reason why we opt for meta-barcoding is that because it's less time consuming and it's more cost effective, and when it comes to identifying them, based on their morphology and using a microscope, we don't have that much taxonomic expertise in South Africa, where for each of the different groups, and besides having taxonomic expertise, it'll be too time consuming to sit under a microscope and go through these thousands and thousands of individuals trying to identify them. And also remember, there's also cryptic species, where some species look the same, but they may be different species, or they look different, or things like that. Cryptic species is the problem. They are very, very tiny and hard to handle. So when you're even trying to take out like a copy pot from a bottle, you could damage its legs, whereas that's one of their morphological characteristics that they use to identify them. So that's why we opt for DNA meta-barcoding, because so we rely on taxonomists quite a lot still to build our reference libraries. So like BOLD and GenBank, we rely on taxonomists to take out single species and be able to extract its DNA and identify these species and add them to these online reference libraries, right? So when it comes to meta-barcode, it makes our lives a bit easier because these species are already present online, but we're able to identify them very quickly and more accurately based on these online reference libraries. So that's one of the reasons why we are using meta-barcoding, because it's very difficult now to actually find trained taxonomists here in South Africa. It's also very difficult to work with these very tiny specimens and be able to identify them using the microscope.
Thank you. Aisha, over to you.
And thank you, Dr. Ash. There's another question in our chat before we go to you, Roland Goetz. So I'm going to ask a question from Greg about, is there a formal global network for coral reef management, a collective strategy where regions coordinate their efforts and assist in what might benefit all from this knowledge? And I think that would be for Justin.
Sorry, yeah, there is, and Dr. Sean Porter, that heads up the coral portfolio at ORI, is part of that and represents the Western Indian Ocean on that platform. He's routinely attending conferences overseas, and there is very much a trend to a collaborative effort, because the impact of global climate change is affecting the broad spectrum. So the coral community is very much getting together from the academics to try and mitigate and look at strategies that would be effective in place.
Thank you. Aisha, it seems to me you've got a bit of connectivity problem there. Roland, I'm going to ask you to unmute, and we'd love to listen to your question. Thanks.
All right. I'm interested in asking the question that can be for probably all marine organisms, is the increase in carbon dioxide and the ocean being a major sink for carbon, and the impact that has in changing the pH of seawater. I think my understanding is that it's alkali around 8, and with the increase in carbon dioxide, an impact of changing the pH become more acidic. What impact is that having, and is it being monitored?
Yes, thanks for the question. So Dr. Sean Porter, that I just mentioned, has been doing an ocean acidification experiment where they've been looking at mesocosms downstairs in the ORI lab and testing predicted levels into the future and looking at how that affects the growth of corals. So that has been a project that started just after I had left and gone across to the aquarium. But there is very much a global trend for understanding how ocean acidification is going to affect the corals. And from a reproductive side of things, it's not just physiologically on what it does to the skeletons, because we know that under high levels it can dissolve the skeletal tissue or the skeleton itself, but it's the compounding effect of that stress, which will then have impacts on the life history stages. So how much energy then actually gets put into reproduction.
Roland, I'm unmuting you again if you want to add on.
Okay, I was thinking about not only corals, but everything. Zooplankton. Obviously, if you change the habitat, the water, and you change the pH of the water, it could have impacts all across the board with everything. Or other things being looked at, maybe coral as an indicator, but what else?
There's also a student that did a foraminifera study, so very small stuff that's found in benthic samples, and she also looked at how ocean acidification affects those. So in the benthos as well has also been investigated at ORI. But yeah, a lot of the research at ORI's applied sciences and the levels of research projects that are looking at ocean acidification, the taxon, I'm not aware of any other projects that are going on at the moment. Don't know if you can add on that.
Just to add on that, also it impacts different species very differently. So as the ocean continues to acidify with a lower pH, there's some species, and it might be across various levels, who will actually start thriving in that kind of environment, and it's then going to negatively impact on various others. So they definitely different species who've got a wider range that's optimal to them. And our first concern would be those in the much tighter range. And of course, the corals are a very good indicator species of that. So it will definitely be a long-term negative impact, I would say, on majority of species in the ocean when you shift out of that optimal range. Ash?
Thanks, Marika. Roland,
so we haven't specifically looked at the effects of this pH and the different changes
We're not.
Are we not? We haven't looked at these on zooplankton yet, but like I mentioned, zooplankton are very good indicators of what's happening in the ocean, and zooplankton are definitely affected by changes in pH, changes in temperature of the water, changes in whatever's going on in the ocean. Zooplankton respond rapidly to what's going on. So if we had to look at the changes maybe in a closed system like an aquarium type, for example, if there was zooplankton in there, if there was changes in the pH, we'd be able to rapidly check the difference in their community composition. We haven't looked at that currently yet.
Okay, thank you. That's answered. I just wanted to put it on the radar screen.
Thank you, Roland. Thanks to the team. Aisha, over to you.
Thank you, Chris. I see Johanna's question.
I think I'm gonna aim my question at Mareike, but you're welcome to pass it along. Mareike, on behalf of Tawani, Nadia, and all the other share screen students who have perhaps completed a B degree in conservation or science and is now wondering, how do I turn my direction towards the ocean? What do they do? How do they get involved?
Oh, that's fantastic. Move to Durban. Eh? So, you know what? Best would be really to join this ocean network, which is, I call it the Ocean Tribe here in South Africa. And they're phenomenal projects. And you know what? All the research we do is actually directed at conservation in how to either utilize marine resources very sustainably, or how to protect what is very endangered. So it's really just you know, through platforms like LCA, make connections with us. We often, I mean, as you often have calls out for either research assistants or postgraduate projects. So you don't have to have an undergrad or even honors in marine science to pursue that as future studies or career. And for those who live along the coast or near any of the bigger aquariums, whether it's Cape Town or here, there are always amazing volunteer opportunities, and keep an eye out for internships, because you're just spending time with this kind of group is already inspiring. And yeah, I think there's huge scope in the ocean research and education space. So I would encourage, you know, all students who are interested in this field to get hold of us, come and visit us, come and see what we do. And you might, like many here, just stay here forever. I mean, Justin's from like literally throughout his master's and PhD and now running the aquarium. So yeah, please stay in touch and come and visit. I don't know if any of you want to add. It's a magic world out there, World Ocean Day.
Thank you. Thanks, Mareike. I really appreciate the presentation.
Thank you. Just to add to that, Yuan, I think it's always speak in share screen Africa that sometimes it's what you know, but very often it's who you know. And this is a great networking platform right here in front of you right now. And we did share the website. So I don't know if Marie just wants to share that website again. And then I'm going to ask Andy Klee to unmute, please.
Thank you very much. Brilliant. What a great team. You're very lucky to have each other, and it's really impressive to see what you're doing. Right, bit of a cheeky question maybe, but it's kind of inspired by Damien Mander and the Akashinga and what they eat in their camps. I don't know if you remember, they've gone vegan, but should we be talking about seafood or sea life? And while I understand right now, some people have to eat seafood, sea life as their primary source of protein and nutrition, quite a few of us have a choice as to whether we do or not. But in the future, if we're serious about ocean conservation, should we perhaps try and move towards a world where we don't see ocean resources, we just see ocean life, and look at different ways of meeting our protein needs and other minerals as well? We've got deep sea mining on the horizon. So traditionally we've done it, but the pressure is so great. Do we need a cultural change? And would that feed into your communications, all that? I mean, have a chat about it anyway. It's a hell of a topic, but I think it's something we should be thinking about if we're serious.
Andy, we should have a chat and a vote. Over to you, Mareike.
I'll kick it off because you know, I operate sort of in the seafood space, but disclaimer, I don't eat fish, I don't eat my friends. However, it's the healthiest animal protein in the world. And Nikhil and I were chatting about it actually earlier today. 20% of animal protein consumed is fish. And fisheries, wild fisheries hasn't increased in many years where aquaculture is taking over. But now, not the problem, but the challenge comes in. And as you mentioned, Andy, is, you know, we're talking about many, many livelihoods that's at stake if we had to say no more seafood. And I actually give a lot of credit to the sustainability ratings, even though I know they also get a lot of flack. But like MSC or our local SASSI is really important. So that's our recommendation to the consumer is, you know, go for a sustainable seafood certification or accreditation at least. And then we need to support through research small scale fishers and the small scale fishery community and subsistence fishing, because the ocean's incredible, you know, and it's really full of resources that we just need to manage better. And there are success stories. If we look at the hake fishery that is now SASSI green, I mean, the Namibian hake fishery also recovered. So with really good management, it's certainly possible. Otherwise, you know, if we don't look after our fisheries and fish resources, and if we don't practice responsible aquaculture, we most likely will have to eat jellyfish, because I think there'll be lots of jellyfish in future. And in the East, they're actually trying to figure out how to create nutritional snacks from jellyfish, or of course, sea plants and algae. And it's interesting in the Western world, that's all the rage and everybody's growing it and talking about it and wanting to eat it, where in the Eastern world, it's been happening for thousands of years, and it's really healthy as well. But yeah, so as a non-fish eating person, I'm actually promoting responsible, you know, consumption of seafood, because it is so incredibly healthy. And what I've seen a lot abroad actually is like plant-based, like fake fish fingers and fake salmon that actually looks like salmon. But I am also inspired by people who just say no more seafood, but then generally it's sort of quite a luxury choice to be able to make. And I know there are thousands, actually millions of people very dependent on their livelihoods in the fisheries industry worldwide. So I always promote, you know, if we look at how we can make an impact, it needs to be really good for people. It needs to be good for the planet. It has to have a purpose. And ultimately, there needs to be a bit of money involved somewhere. Otherwise, nothing works out. And it's just to find that balance. I don't know if anybody else wants to add to that from our side. Yeah. I don't know if you're satisfied, Andy.
Yeah, not 100%. I mean, we'll probably agree to disagree. I mean, I'm, you know, in a very luxurious position in the Western world that I'm in, but I think longer term, I mean, we're talking about a just transition away from fossil fuels for people who are employed in the, I mean, this is sort of 20, 30, 40, 50 year vision, I guess. Just because we've always done it, we haven't. And there are other ways that are less environmentally damaging to meet people's protein needs than by eating other animals, including fish. It's just a way of looking at it. But and I think I'm completely with you. Right now, the work you're doing to make sure what we do take from the ocean and consume is done as responsibly and as least damaging as we can. But part of me thinks it's just with the other pressures on the ocean and the importance of it being a living system, anything that's taking living things out of that environment at the moment is just, you know, it's going to decline eventually. Maybe we're just slowing it down a bit. I don't know. It's a really difficult one. And you know, I think we're all on the same side. It's just, it's kind of, you know, you've got to pick away at these things sometimes. So, but thank you ever so much, and brilliant work, everybody. I'm super inspired. Yeah, you're all my favorites tonight, so thank you very much.
Before you go, Andy, before you go, but I just add something. I don't think that the issue is necessarily fishing. I think it's overfishing. Yeah. And so it depends on the species. And so you based in the UK, from what I remember.
Yeah.
Yes. So in South Africa, we have the South African Sustainable Seafood Initiative. It's called SASSI. And like the fish that I worked on was kingklip, and we've seen it move from an orange designation, which means it was fairly unsustainable, to a now green designation. And this happened like in the past 7 years because of management practices in that. So I don't know if you have a platform like SASSI in the UK.
I don't know enough about it, but I do know, you know, our fish stocks, you know, you have the wonderful sardine run. We used to have a herring run here, you know, millions of fish swimming by, local communities supported by it. And in the early 70s, the stocks completely crashed. And there's one guy in a small village who's the last herring fisherman. You know, we completely blew it here. And every time you think, oh, well, you know, let's get some more marine protection in, the fishing industry's up in arms because whatever little bits left, they want to get at, you know, and there's economics and shareholders and all that jazz. I'm not sure about that wider management thing. We have a Marine Conservation Society who produce a sustainable fish guide. But who knows who, you know, it's like all these things, there are other interests and they're trying to balance a lot of, you know, difficult things.
So the global certification body is the Marine Stewardship Council, and they assess fisheries all over the world. And actually one of our senior scientists is an auditor. So they'll audit the fishery to look at, is it a sustainable stock? Does the fishery method have an impact on the environment? So there are actually 5 aspects that they assess. And then it will be graded in similar to SASSI, whether it's like red, orange, or green, but just a slightly different grading. And there's various ones all over the world, and aquaculture's got the same. So the Aquaculture Stewardship Council. So I feel it's at this point in time, it's quite well researched and maybe not that well managed, because it's very difficult to have observers on all the ships and make sure there's not, you know, it's not overfishing, which is still happening. But we can confidently state that there are healthy fisheries. There are definitely stocks that is sustainable. However, there are many that have completely commercially collapsed, and there are many sort of on the edge. So as long as governments actually manage what's in their, you know, exclusive economic zones really well, then it should be something that we can manage. But if you look at the marine traffic app, it's always mind-blowing to me, because I mean it's so busy. And then all the fishing vessels are listed on there in their specific color, and they're all registered. And then there's sort of unidentified, unregistered, and I call them all the pirates and the poachers. Yeah. And there are quite a lot of them. And that's where the problem lies, you know, it's just taking too much. But yeah, I think there are definitely too many livelihoods at stake, and the sea is amazing. It's somehow can still deliver. And I want to give the ocean a lot of credit in saying it's going to bounce us off here and there, you know, every now and again. But at least there's so much awareness. I think we're more aware of our impact on the ocean, and we're more aware of what we can do to try and maybe not correct it. That will take a very, very long time, but to minimize or reduce negative impact than ever before. And that to me is like very hopeful. And that's probably the reason why we all do what we do, because we still have hope.
Oh, brilliant. Well, Maria, I'm gonna give you the ultimate compliment. You've echoed the words of Dr. Sylvia Earle at the UN conference earlier on today. I checked into the live broadcast of that, and that was pretty much, we've never known more, and we've never known how important it is to look after it. So I'll on that high, we'll get on to the next question. Thank you ever so much.
And Andy, you must note that in the chat, Stephen Blinkensop, if that's how you pronounce his surname, just wrote very thought-provoking viewpoint. Thank you, Andy. So really something to think about. Aisha, I think we should start closing down and maybe give Tawani the last question, if you're okay with that. All right.
Let's go.
Tawani, we're going to unmute you. There we go.
Hi, sorry, thank you. I do have one question. When I was working with the NGO in the Mediterranean area, the biologist said that at the moment, at least in that area, the fish that you can see are all basically juveniles. So you don't have the older fish in the area anymore. I don't know if that's the same situation in South Africa because of overfishing, because I don't really know the situation in South Africa at all. But do you think that it's the same, or maybe a little bit older, some older species at least?
So that's a very relevant question, and thanks for that. And I actually spent quite a bit of time in Europe and had a good look at the fisheries along the North Sea, and you see exactly the same issue there. And I was surprised because I went to various fish processing establishments, and some of the like flounder and sole were like this size. I could not believe that that was still being processed and sold because there's such a high demand. So it's very much species dependent. A lot of species have adapted, which is actually quite sad, but amazing at the same time. For instance, just the sardines, are reaching sort of sexual maturity much earlier than, let's say, 2 or 3 decades ago because of the pressure on the stock. So, yeah, like I said, that's quite amazing, but also sad. And I think in South Africa, our scientists and many of our RE scientists also contribute to stock assessments and making those decisions. So for a lot of, our lionfish, for instance, you've got size limits. So you're not allowed to catch or bag fish below a certain limit, and sometimes above a certain limit as well, if it's like really good broodstock quality. And what has happened in the past with, for instance, the dusky cob or kabeljau as together, it was dusky cob and silver cob. Nobody knew they're 2 different species. And dusky cob mature very, very late. So they're actually at a very big size, where silver cob mature really early. So the size limit was based on the data from silver cob, which meant that the dusky cob were caught before they were sexually mature, and the whole species fishery collapsed. I mean, they're less than 2% of pristine stock of dusky cob left because of basically poor guidelines, which led to overfishing before they reach maturity. So a very, very valid comment. I would have to go and read up a bit more about exactly how we deal with it with our different fisheries. But also, like an interesting thing, for instance, I&J's is one of the oldest, biggest fishing companies in South Africa, and they now only catch hake. And with the hake, they get certain bycatch, allowed bycatch, but they're not catching sardines or pilchard anymore, or various other species that they used to catch, purely because it's not sustainable and they can't get their quotas. So in South Africa, the fishing companies are permitted to catch a certain amount. So they get a quota, which is calculated through stock assessments. And the scientists will make recommendation to the fisheries department and say, the stock can only carry this amount of total catch, otherwise it will reduce or collapse. So for long-term, I suppose, security of a business, these fishing companies have to adhere to that. And then in South Africa, a lot of these fisheries vessels have got independent observers on board to make sure that they stay within their quotas and their permit conditions.
Thank you so much.
Thank you,
That was
Marika.
fascinating.
Tawani, you okay?
Yes, thank you so much. That was great.
Thank you. Aisha, ladies and gentlemen, I think it's now time for us to say goodbye. I do want to just ask, firstly want to say to the SAAMBR team, again, your passion is just absolutely incredible, and your dedication and your hard work. And thank you for a lovely debate and discussion too. And Andy, thanks for stirring the pot a bit. We shouldn't just think about today, we've got to go into the future. I don't know if that fish behind you is there deliberate or every day. Anyway, to the whole team who is behind all this, and then the team of SAAMBR, and also the audience, I thank you for your involvement and your concern and your interest. And we wish you all the best